So we have already started enzyme chapter. So we are in classification of enzymes. You know that enzymes are biocatalyst. Okay. So I quit a few slides because it's already over. So there are classification enzyme oxido reductase, transferase, okay, hydrolases. These are the hydrolases, example for hydrolases. Okay, so next we'll see fourth classification of enzymes. Okay, fourth classification of enzymes, that is lyases, lyase. These catalyze the splitting of molecules by mechanism other than hydrolysis. Hydrolysis mechanism means enzymes are called as hydrolases. So they are splitting of molecules other than mechanism other than hydrolysis. Okay, see, a b plus xy gives ax plus by. Example for this lyases, aldolase, fumarase, these are the enzymes comes under metabolism. Okay, example 1, fructose 1,6 bisphosphate gives glyceraldehyde 3 phosphate plus dihydroxyacetone phosphate. Aldolase is the enzyme. Aldolase enzyme which convert fructose 1,6 bisphosphate to glyceraldehyde 3 phosphate plus dihydroxyacetone phosphate. Here see, fructose 1,6 bisphosphate is 6 carbon. Okay, which is 6 fructose, you know, it is a monosaccharide, glucose fructose monosaccharide. Fructose is a keto sugar, so fructose is hexose, 6 carbon. Here see the reaction, example 1, 6 carbon is split into glyceraldehyde 3 phosphate which is 3 carbon plus dihydroxyacetone phosphate which is also 3 carbon. So 6 carbon is split into 3 carbon. So this lyases catalyze the splitting of molecules by mechanism other than hydrolysis. Okay, so this is the one example. Second example we can give here also. I remember, student, most of them gets tensed by seeing the formula. No need to write the formula, only write in the word, like pyruvate plus pyruvate gives acetaldehyde plus carbon dioxide in the presence of pyruvate carboxylate. See here what happens. Pyruvate gives acetaldehyde, CHO group is there, it's aldehyde, and it there is a removal of carbon dioxide from pyruvate. Okay, so formation of acetaldehyde and carbon dioxide. So pyruvate is splitted here also. Enzyme, this is example for enzyme pyruvate carboxylase is comes under enzyme group lyase. Okay, so pyruvate is split to acetaldehyde plus carbon dioxide. From the pyruvate, carbon dioxide is removed, okay, by the enzyme pyruvate carboxylase. So first example you seen, fructose 1,6 phosphate to glyceraldehyde 3 phosphate plus dihydroxyacetone phosphate. Aldolase is the enzyme. So second enzyme is, you can give the example is pyruvate decarboxylase. Both are lyases. Next, fifth class is isomerase. Enzymes involved in all isomerization reaction. Enzymes involved in isomerization reaction. So you know that what are isomers, having the same structural formula, same molecular formula but different structural arrangement. Okay, same molecular formula but different structural arrangement, they are isomers. Isomerases are enzymes involved in isomerization reaction. Okay, change of isomers, like glyceraldehyde 3 phosphate converted to dihydroxyacetone phosphate. These are interchangeable. Okay, dihydroxyacetone phosphate also can be converted to glyceraldehyde 3 phosphate. These names and all comes under glycolysis. Okay, this is one of the middle step of glycolysis, breakdown of glucose. Glyceraldehyde 3 phosphate is converted to dihydroxyacetone phosphate. Okay, so enzyme is triose phosphate isomerase. See the enzyme name given, 3 carbon triose phosphate isomerase. This glyceraldehyde and dihydroxyacetone 3 carbon, so they named as triose phosphate isomerase. So racemases, epimerases are also example for isomerases enzyme. Okay, so one more example, glucose isomerase, conversion of glucose to fructose, glucose isomerase. Okay, so again the structure not required. Glucose converted to fructose, reversible reaction, glucose isomerase is the enzyme. So before example is triose phosphate isomerase, one example. Second example for isomerase is glucose isomerase. Like that many examples we can give. So next is last one, ligases. Ligases you have seen. What is ligases? No confusion here. Ligases, splitting of molecules by mechanism other than hydrolysis. Ligases, LYA. Here you see this is ligases, LIGASES, catalyze the joining of two compounds with the energy provided by ATP molecules. Remember that ligases, ATP is utilized. What is ATP? Adenosine triphosphate, energy rich molecule. Adenosine triphosphate, it is a nucleotide, remember. So ligases catalyze the joining of two molecules with the energy provided by the ATP molecules. Here ATP is used because in this reaction, remember that ATP is converted to ADP plus EI. That is all. So there is no confusion with ligases and liases. Liases, splitting of molecules by mechanism other than hydrolysis. Ligases means catalyzes the joining of two compound with the help of ATP. From where ATP will get? By energy will get from ATP. Here ATP is used in ligases reaction. ATP is used. For example, acetyl CoA plus carbon dioxide plus ATP, malonyl CoA plus ADP plus PI. ATP is converted to ADP. Okay, pyrophosphate. PI means pyrophosphate. So here joining of two compound, acetyl CoA plus carbon dioxide to give malonyl CoA. ATP is converted to ADP. Acetyl CoA carboxylase is the enzyme. So acetyl CoA carboxylase comes under enzyme ligase. Okay, so is acetyl CoA carboxylase is example for ligase, enzyme ligase. One more example is second example is glutamine synthetase. See, this group of enzymes are called as synthetases. Glutamate is converted to L-glutamine. You see the amino acid, that is L-glutamate, MATE, is converted to L-glutamine, MINE. Glutamine synthetase is the enzyme. Okay, enzyme involved. Glutamine synthetase is the enzyme involved. Okay, so here again joining of molecule is there, joining of two compound. Okay, so ammonia is added in glutamine. Okay, so so glutamine synthetase is example for ligase. First example we can give acetyl CoA carboxylase. So you have already seen 6 classes. What are those classes? What are those classes? Oxidoreductases, transferases, hydrolases, then liases, isomerases and ligases. Now let us see factors affecting enzyme activities there in your syllabus. So what are the factors which affect enzyme activity? Enzyme concentration first of all. Okay, enzyme concentration, substrate concentration, pH, okay, these and all, temperature, okay, these are mainly 4 factors which affect enzyme activity. Enzyme concentration, see, when sufficient amount of substrate is present, rate of reaction is directly proportional to enzyme concentration. So what we expect, there is a straight line. When sufficient substrate is present, that word you have to remember, when sufficient substrate is present, the rate of reaction is directly proportional to enzyme concentration. Rate of reaction is directly proportional to enzyme concentration. So you should get straight line. Okay, see, enzyme concentration in x-axis, enzyme velocity is y-axis. So when sufficient amount of substrate is there, rate of reaction goes on increases. When sufficient amount of substrate is there, okay, enzyme concentration, enzyme when sufficient amount of substrate is the rate of reaction is directly proportional to enzyme concentration. Okay, when substrate is there, more and more enzymes bind with the substrate, so reaction goes on increases. Okay, so you will get a straight line here. See, second, substrate concentration. First one enzyme concentration, second one substrate concentration. Increase in substrate concentration increases velocity of reaction initially, but curve flattens afterwards. First you get like straight line, then curve flattens. Okay, so substrate, what is the effect of substrate concentration on enzyme activity? E and Z means enzyme. Okay, so increase in substrate concentration increases velocity of reaction initially, but curve flattened afterwards. So you don't get straight line. V max is velocity maximum, we call V max. So here we don't get straight line. Okay, beginning it increases. Okay, increase in the substrate concentration increases the velocity initially. Okay, but curve flattens afterwards. You see substrate concentration x-axis and initial reaction rate on y-axis. See, increase in substrate concentration here increases velocity of reaction initially. See, you can see there half Vmax Km and all, half Vmax up to certain concentration. Increase in the substrate concentration, velocity also increases. After that, curve flattens. Why? Because there is no more enzymes to react. Increase in the substrate concentration, because enzyme concentration is constant there. Okay, so there is no more more and more enzymes get reacted with the substrate. So reaction rate goes on increases. At a certain point, almost all substrate is saturated with enzymes. Almost all enzymes, almost all, at a certain point, almost all enzymes get saturated with the substrate. So there is no more reaction, increase in the reaction goes steady. Okay, after that, that is the meaning. Increase in substrate concentration increases velocity of reaction initially. That is why you can say straight line initially, but curve flattened afterward, because at a certain point almost all enzyme is occupied by substrate, so rate becomes constant, or becomes constant. That is called velocity maximum, is the v axis velocity maximum there. Okay, so before curve substrate concentration, we already said, as substrate concentration is increased, velocity is also correspondingly increased in the initial phase, as I told, but curve flattened afterwards. The maximum velocity obtained is called Vmax. Vmax is nothing but velocity maximum, represent the maximum reaction rate attainable in presence of excess substrate. That is the maximum. Okay, that you can see there. Vmax is written in y-axis at the top. Okay, on left side this you can see V max at the top, that is the maximum velocity attainable. Okay, so this is the action of substrate, this is the substrate factors, substrate effect of substrate concentration on enzyme activity. Okay, so here we get this type of curve. Okay, initially straight, then flattened curve, but in enzyme concentration we get straight line. Okay, so next is this half Vmax is also called half the maximum velocity is called, actually half the maximum velocity is called Km value. What is Km value? You will see next. Half the maximum velocity is called Km value. See here the substrate, this is the same one, continuation. There in substrate concentration, according to Michaelis theory, there is one Michaelis theory, the formation of enzyme substrate complex is reversible reaction. How to show reversible reaction? Arrow mark on both sides. Yeah, E plus S, yes. So formation of enzyme substrate complex is reversible reaction, while breakdown of complex to enzyme plus product release, that is irreversible. Okay, so what is Km value? Remember that Km is a constant. Okay, Km is a constant. What is Km value? Km is a constant. So what is Km value? Km value is the substrate concentration at half maximum velocity. You have to read this 3-4 times, split it and read, you will understand. Km value is the substrate concentration at half maximum velocity. V50 we call, see this curve, it is there, half Vmax. Vmax means velocity maximum. Half velocity maximum is called Km value. So Km value is substrate concentration at half maximum velocity. You read, look at this curve and read, okay, 2-3 times, you will understand. Km value is substrate concentration at half maximum velocity. Vmax is the maximum velocity. Half Vmax is, or 50 also we call half maximum velocity is Km. Okay, Km value is substrate concentration at half maximum velocity. So it denotes 50% of the enzyme molecules are bound with the substrate molecule at a particular substrate concentration. So what Km value denotes Km value is the substrate concentration at half maximum velocity. That is the definition for Km value. So Km value denotes that, it indicates that 50% of enzyme molecules are bound with the substrate molecule at a particular substrate concentration. So it is the signature for each enzyme. Each enzyme have different Km values. Okay, Km or Michaelis Menten constant, they derived it, so it's called Km or Michaelis Menten constant. No need details for you. You should know what is Km value definition and what is the significance of Km value. So Km is substrate concentration to produce half maximum velocity. So here again this is repetition. 50% of enzyme molecules, MOL apostrophe S is molecules. Okay, 50% of enzyme molecules are bound with the substrate molecules when substrate concentration equal to Km value. So V is equal to Vmax S divided by Km plus substrate. Okay, that if you remember or need not remember, but you should know what is Km value. Then substrate concentration to produce half maximum velocity, half maximum velocity. Okay, that is the definition for Km value. Why this Km value is important? There is some importance in significance of Km value. At constant, it's a constant. Km is the measure of affinity of any enzyme for its substrate. There are various metabolic reactions. So we can measure the affinity of enzyme for its substrate by looking at Km value. It is a characteristic feature of a given enzyme are termed as signature of the enzyme. This word you have to remember. So Km value is termed as signature of the enzyme, used to measure the strength of enzyme substrate complex. So decrease in Km value, that is arrow mark down, okay. Decrease in Km value shows a strong affinity between B/W means between enzyme and substrate. So you see this last sentence, decrease in Km value, lower Km value indicates a strong affinity between enzyme and substrate. If Km value is less means there is a strong affinity between enzyme and substrate. Why? Because see the definition. You can see the definition again, go back 2-3 times, see the definition, okay, you will understand. Km value, okay, is substrate concentration at half maximum velocity, okay. So decrease in Km value shows a strong affinity between enzyme and substrate. This is one example given in this slide, okay. Glucokinase and hexokinase are enzymes which phosphorylate glucose to glucose 6 phosphate. This is comes under glycolysis, okay, this reaction. So both are enzymes. Hexokinase exclusively for tissues, glucokinase exclusively for liver. Glucokinase is present in hepatocyte, means in the liver. Compared to hexokinase, glucokinase have higher Km value, lower binding affinities. So Km value of hexokinase is high compared to glucokinase. So hexokinase is found in most tissues. Compared to glucokinase, hexokinase has lower Km value, okay. Hexokinase has lower Km value, glucokinase have more Km value. Due to high affinity, if Km, you see the significance there, low Km value means high affinity. So due to high affinity, because of low Km value, glucose is utilized by hexokinase even at low concentration, because hexokinase have lower Km value, so it is having higher affinity to glucose. So due to high affinity means low Km value, glucose is utilized by hexokinase even at low concentration in the tissue, whereas glucokinase act only at higher glucose level, that is after meal, because excess glucose is converted to glycogen by enzyme glucokinase. Hexokinase act in lower blood sugar concentration also, because it supplies energy to tissues, okay, that is the meaning. So next factor is factors affecting enzyme activity is temperature. No need to see much here. Here you will get a curve type temperature. Rate of reaction increases with increase in the temperature up to a maximum, then it declines. You know that each enzyme has its own optimum temperature. The temperature at which maximum amount of substrate is converted to product unit time is called optimum temperature. Each enzyme has its own optimum temperature. So you get a bell shaped curve here, okay. So usually it is between 40 optimum temperature is between 35 degree. Most of the enzymes act at best at 37 degree, 35, 36 or 37 degree, okay. Next, pH. Increase in hydrogen ion concentration influences the enzyme activity. pH means hydrogen ion concentration more means it is acidic, you know that. Example is given, pepsin, pH 1.2, it acts best at pH 1.2. pH of acid phosphatase, ACP means acid phosphatase 4.4 to 5, 10 to 11 for alkaline phosphatase. pH high, as the name indicated, alkaline phosphatase 10 to 11. So here also same type of curve, okay, bell shaped curve. Each specific enzyme can only work over a particular pH range. Each enzyme has its own optimum pH, like temperature, where the rate of reaction is maximum. Here is given some of the enzyme and this curve ABC, A for amylase, B for pepsin, C for lipase. You can see maximum amylase optimum pH is 7.2. It acts best at 7.2. Pepsin act best at pH 2. Lipase act best at pH 9. Okay. Changes in pH can affect ionic and hydrogen bond responsible for specific tertiary shape of enzymes. Extremes of pH break these bonds and denature the enzymes. Okay. So same bell shaped curve will get for pH and temperature, optimum pH and optimum temperatures. There are other factors less important. Effect of product concentration. Accumulation of more and more product decreases the enzyme activity. If more and more product already is there, then enzyme activity decreases. Then some activators also. Some of the enzymes require certain inorganic metallic ions like magnesium, calcium, cobalt and all for their optimum activity. So metal activated enzymes means here the metal ion is tightly held with the enzyme. Enol is required magnesium. Enol is one of the glycolysis enzyme which required magnesium. This is one example. Some enzymes are called metalloenzymes. Here metal is tightly held by the enzyme. Cytochrome oxidase is require iron and copper. Cytochrome oxidase is a electron transport chain enzyme which require iron and copper. Metal activated enzyme, what is the difference between metal activated enzyme and metalloenzyme? Metal activated enzyme, metal is tightly held by the enzyme. Metal is not in metal activated enzyme, metal is not tightly held by the enzyme. Okay. In metalloenzymes, here the metal is tightly held by the enzyme which cannot be removed easily. That is called metalloenzyme. In metalloenzyme, metal is tightly held by the enzyme. These metals are iron, copper and all. Okay. So this is the effect of activators. Means these cations or metals which further enhance the reaction. Some enzymes require these certain inorganic metallic cations. Okay. Next is coenzymes. What are coenzymes? They are non-protein part of the enzymes. See, enzyme as it is hollow enzyme we call. Protein part is apo enzyme. Non-protein part is called coenzyme. Non-protein part we call coenzyme. Enzyme is hollow enzyme. Protein is apo enzyme. Non-protein part is called coenzyme. See there right side non-protein word comes together. Okay, please remember there non-protein word comes together. That is comes next sentence. Enzyme is hollow enzyme. Enzyme is called protein part of the enzyme is called apo enzyme. Non-protein part of the enzyme is called coenzyme. Okay. So non-protein part, that is prosthetic group of the enzyme is called coenzymes. What are coenzymes? Non-protein part of the enzymes are called coenzymes. They are with lower molecular weight, heat stable. They are required for biological activity. They are loosely bound to enzymes. Released from apo enzyme. From the enzyme, apo enzyme means from the enzyme, protein part of the enzyme after completion of the reaction. But one important point you have to remember that most of the coenzymes are derived from B complex vitamins. We are studying some of the B complex vitamins. Okay, niacin, riboflavin, all those things, biotin and all. So most of the coenzymes are derived from B complex vitamins. Okay, these are very important. Remember, coenzyme is one of the most important short note, either 3 or 5 marks. So you have to write these properties also. The protein part of the enzyme gives the necessary three-dimensional infrastructure for chemical reaction, but the group is transferred from or accepted by the coenzyme. Inside the body, when the reaction is complete, the coenzyme is released from the apo enzyme and can bind to another enzyme. See, remember that one coenzyme can bind with the second time. Once the reaction is completed, the coenzyme is released from the apo enzyme and can bind to another enzyme. So, one molecule of the coenzyme able to convert large number of substrate molecules with the help of enzyme, because After the completion of the reaction, they release it from the apoenzyme and can bind to another enzyme. So, one molecule of the coenzyme is able to convert a large number of substrate molecule with the help of enzyme. Most of the coenzymes, as I told, they are derived from B complex vitamins like niacin, riboflavin, uh, all, all this, pantothenic, folic acid and all. Example for coenzymes is given. See, you can remember just ATP is a coenzyme, okay, adenosine triphosphate, group transferred is phosphate. Pyridoxal phosphate, PLP, is a coenzyme, group transferred is amino group. Biotin, group transferred is carbon dioxide. Coenzyme A, group transferred is acyl group. NAD, a group transferred is hydrogen and electron. Their electron should come along with the hydrogen. Remember last sentence, NAD group transferred is hydrogen and electron. Okay, no confusion. There is a two column. One is coenzyme, second one group transferred. ATP transfers phosphate, PLP transferred amino group, biotin transfers carbon dioxide, coenzyme A transfers acyl group, NAD transfers hydrogen and electron. Electron should come along with the hydrogen. Okay, last sentence. So cofactors, there are one more term, coenzyme, non-protein part of the enzyme, remember, or prosthetic group. Protein part is apoenzyme, non-protein part is coenzyme. What are cofactors? These are collective term we use. Coenzyme plus metal ions means non-protein part of the enzyme plus metal ion together called cofactor. So coenzymes comes under cofactor. Remember, coenzyme comes under cofactor. There is no confusion. Coenzymes also comes under cofactor. It is a collective term. So organic cofactors are called coenzymes. Okay. What about inorganic cofactors? They are called activators. So what are coenzymes? They are organic cofactors. One more term for coenzymes. Coenzymes are organic cofactors. So it is a cofactor means it is a collective term used to include coenzyme plus metal ions. Cofactors may be organic or inorganic. If it is cofactor, if cofactor is organic, then what it is called? If cofactor is organic, then what it is called? Coenzyme. If cofactor is inorganic, then it's called activator. No confusion, you have to read it twice. So activators, inorganic cofactors necessary to enhance the enzyme activity, calcium, magnesium, manganese ions. Metalloenzymes means enzymes which require certain metal ions for activity. Copper is a metal. This is required for the enzyme tyrosinase. This is the one example. Ion activated enzymes, without the metal ions, enzyme may be active, but reaction rate is less. But the metal ion is added. If metal ion is added, the activity is enhanced. So without the metal ion also, enzyme may be active, but if you add metal ions, some enzymes are activity is increases. For example, calcium activate pancreatic lipase. Okay, otherwise activity of pancreatic lipase is very slow. Calcium ion which activate activity of pancreatic lipase enzyme. Okay, so metalloenzyme, enzymes which require certain metal ions for their activity. So all this you can include under cofactor. Metalloenzyme, there is ion activated enzymes. Without metal ion, this is just a repetition of this ion activated enzymes. Okay, these are the things. So here we have to remember that coenzyme is important. Examples also you can give and properties also you can give. These are the properties of coenzymes, and most of the coenzymes are derived from B complex vitamins. Okay, that is all. Okay, thank you. This is just repetition of previous slide.